Hi everyone, my name is Ayush. I'm going to be talking to you today about the sex and disease-specific differences in lipolysis. So, obesity-associated type 2 diabetes typically stems from adipose tissue dysfunction, which basically means that there's problems with the fat cells in your body. And so what we see in particular is the dysregulation of adipocyte lipolysis. So lipolysis is the breakdown of triglycerides into its separate components. And these components consist of one molecule of glycerol and three free fatty acids. And so what can happen after lipolysis is all of these molecules can be released into the bloodstream, and that can be very problematic since it contributes to insulin resistance, which is a very common problem with type 2 diabetes. So our objective was essentially to see how diabetic status as well as sex plays a role in the expression of lipolytic signaling molecules. So basically how those two factors play a role in affecting lipolysis in individuals. So the way that we sort of went about this was we first took fresh, whole visceral adipose tissue. So the reason we look at visceral adipose tissue is because this is essentially the fat that's around your gut and your abdomen. And this is the one that's typically more associated with diabetes and can become problematic. The other type of fat tissue that we can look at is subcutaneous adipose tissue. And that's basically what lies just below your skin, but that's typically not as problematic in type 2 diabetes. And so we take the whole VAT and we'll cut it up, put it in a collagenase solution that will help digest and break it down. And then from there, we'll filter it out to get rid of the extracellular matrix and the things that we don't want. And that will essentially leave us with the primary adipocytes, so the actual cells that we want to work with and do the experiments. And from here, we'll separate the primary adipocytes into two separate batches. So one of them will be for RNA extraction, we can do things like PCR, and then the rest of the adipocytes we will plate and use for lipolysis experiments. So what you can see in image B here is basically a fluorescent microscope image of live primary adipocytes. And so the blue indicates the nuclei of the cells and the green indicates the lipids. And so what we found in our experiments was essentially that when you stimulated cells with isoproterenol, this increased the lipolytic response of the cells. And so isoproterenol is a beta adrenergic receptor agonist. And this basically causes the lipolytic responses in cells to increase. And so seeing an increase in lipolysis in these isoproterenol stimulated cells makes sense. That's what we expected. But what we additionally found is that in particular, there was an increase in the diabetic cells that were stimulated with isoproterenol compared to the non-diabetic cells. And so this basically just tells us that patients with diabetes will typically be seeing greater rates of lipolysis than patients without diabetes. But what we also found when we further stratify the data by sex is that the key finding was that female cells, female diabetic cells in particular, when stimulated with isoproterenol, showed the greatest increase in lipolytic response compared to all other sort of controls. And so we see, particularly in figure 5B here, what we see is that this pink bar in the very middle, that shows the diabetic female cells that was stimulated with isoproterenol. And we can see that their lipolytic response was much greater than any of the other factors. So after this, what we did was when we looked at RNA sequencing was try to sort of see if there was a difference in the lipolytic gene regulation between diabetic and non-diabetic adipocytes, but we didn't see any significant differences. There was some upregulation. So if you look at ADRB2 or ADCY7, there was a bit of upregulation, but no significant differences. And so when we take all of this information into account, some of the key takeaways are first and foremost that there are indeed diabetic specific effects in the way that adipose tissue has lipolytic responses. But in particular, what we found is that diabetic women have an increased lipolytic response in their visceral adipose tissue compared to diabetic males. And so this is very important because what it suggests is that there is some sort of dysfunction at the beta adrenergic receptor level in diabetic males or in obese males at the very least. And this is basically causing a lack of lipolytic response to isoproterenol stimulation. And so that's our biggest takeaway from what we sort of found. And so moving forward, some things that we can look at in particular are investigating sex and disease differences in forskolin stimulated lipolysis. And so this would be interesting because forskolin stimulates cyclic AMP, which is a different factor sort of along the lines of the complex metabolic pathway. So we could look at that. We could also look at the effect of metformin, which is a very common type 2 diabetes medication, and what that sort of does with the lipolytic responses in patients. And finally, we could also look at where pre-diabetic patients sort of fall along the scale of lipolysis, since they're sort of in the middle ground of non-diabetic patients and patients that are officially diagnosed with diabetes. So that is all I have for you today. Thank you for coming by.